I started noticing differences in him around 2 to 3 months, but nobody else believed me. And they kept pushing it off, telling me that I was just paranoid or first-time mom jitters, and just to accept him for being a little slower on his milestone road. It wasn't until 13 months that he started having seizures. He was diagnosed with infantile spasms at 13 months. Ben has intractable epilepsy. We've tried at least 13 different ASMs. He has an infectious giggle. He is completely nonverbal. He used to say mama and dada before the seizures started, but that never came back. He has a smile that will reach your heart. He has severe neuromuscular issues. You could see the picture there of his spinal fusion surgery. That was an 8-hour-long surgery from T4 to L4 to fix his 71-degree curve. He was diagnosed with osteoporosis at age 10. He's a serial hugger. And if you've met him and you've gotten a hug from him, you will never forget it. Ben thinks farts are funny, which they are. I think we all agree. He has feeding issues. He needs a G-tube. And he's also everyone's favorite person. Ben has the amazing ability to win people's hearts without saying a word or even making eye contact. He is the definition of love and truth. He just celebrated his 13th birthday, and that in itself is a huge accomplishment given how sick he was as a baby and the chances that the doctor said he would make it to his second birthday. When Ben was diagnosed with an SCN2A-related disorder in 2012, and I learned that there were no treatments, no cure, and almost no research being done into this disease due to it being so rare, it was abundantly clear what I needed to do. If we don't build the ecosystem of research readiness, no one will. So what is research readiness? Being research ready translates to actively empowering research and advancement. Being research ready means being prepared and equipped to engage in research activities. It implies having the necessary resources, infrastructure, expertise, and support systems in place to facilitate research efforts effectively. By being research ready, your organization is actively enabling and facilitating research initiatives, and this leads to progress and advancements in the field. So essentially, the phrase is emphasizing the importance of being well prepared for research endeavors and the positive outcomes it can bring. Why should you pursue research readiness? Well, I mean, it's pretty obvious. If you want to get to a cure, you have to be organized. It fosters the generation of evidence and development of an ecosystem, and that's conducive to advancing the understanding, the care, and the outcomes associated with a specific disease state. It encourages the production of data, research findings, and information relevant to the disease. This evidence can be crucial for informing decision-making, treatment strategies, and policy development. In essence, being research ready is critical to promoting research, fostering collaboration, and ultimately improving the outcomes of those affected by a certain medical condition. When should you start working on being research ready? Now, right away, right as soon as you start. If you're in this room or you're listening in virtually, I'm not sure if there's a virtual component, okay, then you have already started because you're here, you're listening, you're trying to figure it out. It's critical to start right away, as no one suffering from a rare disease has any time to waste. So Sean has put this deck together in a really interesting way. I think it's very helpful to think and to look at these 4 categories, these 4 buckets, and trying to figure out where your organization falls. I'm speaking directly to patient advocacy groups in this part. So each organization is on their own journey towards research readiness, and this talk will frame steps you can take, actionable steps, no matter where you fall within these 4 general buckets. So if you're just beginning, you're organized but still figuring things out, you're established, or you're mature. And I'd like to emphasize that these categories are fluid, they're not rigid, and everyone's a little different, and that's okay, and you might be between two of them. Can I just get a raise of hands if of anyone that is a patient advocacy group in this room? That's good, thank you. It helps me to frame. Keep your hand raised if you're just beginning. If you're organized but figuring things out. If you feel like you're established. How about mature? Sounds good. So we have a little bit of everything. So if you think, I think it's easiest to think of research readiness in 4 pillars. You've got clinical phenotyping, basic research, translational research, and clinical trial research. Each of these pillars work together and play off each other to culminate the creation of an ecosystem. Each pillar is crucial for the ecosystem's progress towards research goals. We're going to walk through each, what they mean, the benefits to this work, and how you can make progress depending on which bucket you fall in and where your organization currently falls. Clinical phenotyping. It entails meticulously describing, documenting, and disseminating information about a disease with scientific precision, aiming to develop a thorough comprehension of the clinical presentation. The scientific rigor here is critical. It deepens the insights into the disorder and propels ongoing research and interventions. These publications, they're gonna serve as the cornerstone for subsequent research. And they play a pivotal role in establishing the baseline parameters for clinical studies and facilitating pathways for drug approvals. Clinical phenotyping examples of steps that you can take depending on where you are in your journey. If you're just beginning, talk to your doctor, your child's doctor, your doctor, about getting something into the literature. Ask them to do a case study. If you've launched, but you're still figuring things out, build your community and start to systematically get information about your disease. Empower your community. You can do that through education and through support. Develop a contact registry. If you're established, expand on everything you've already accomplished and diversify. Start collecting natural history study data, whether it be patient-reported, clinician-reported, or data mined, like claims and EMR. If you're mature, enrich and validate that natural history study data. Aggregate the natural history study data. As you know, a lot of times it's siloed. Do your best to try to get it all into one place and make it shareable. Tailor your natural history study collection to capture emerging needs and goals of your community. The next pillar is basic research, which is the pursuit of a deeper understanding of the biology, physiology, and mechanisms of action of the disease. And the benefits to making sure that you get this basic research right, it creates a knowledge base to support clinical care, translational research, and drug development. And just as importantly, your basic science research network will also be invaluable as in your ecosystem. I have to mention, and I might not be the most popular person after that I mention this, but there's a growing trend to bypass this pillar. And we feel like that could be a mistake. In our organization, we call treatments shots on goal. As we all know, drug development is risky, especially in rare disease. And when a drug fails, as many will, we need the foundation of the basic research to evaluate why it failed and quickly turn that into a learning. We're also watching our kids get worse and die as we work towards treatments and cures. So we understand that urgency, but we believe that you can still accelerate the pace of rare disease treatment development while ensuring that rigorous scientific standards are maintained to safeguard the patient safety and efficacy. Balancing that need for speed with the requirements for robust evidence, it remains a critical consideration in this evolving landscape. Okay, here are some examples of things that you can do depending on which bucket you fall in the basic research pillar. If you're just beginning, learn how to read a scientific publication. Google what you don't know and email those authors. Start your network. Introduce yourself to scientists that are working on your topic or adjacent topics. Like, for instance, SCN2A-related disorders is epilepsy, autism, intellectual disability. So I don't focus just on people that are studying SCN2A, but anything in those categories. Begin building a research network. Put together a tracker to make sure that you stay in contact with the researchers on a regular cadence. Set up a Google alert on your computer or your phone for anything that might come up on your disease. Search PubMed regularly. If you're launched but still figuring things out, empowering your community starts right now. Continue building that network and strengthening the relationships. Assemble a scientific advisory board to help you evaluate and vet research. Utilize your platform to amplify the research message. Get on the socials. Create opportunities for discussions. Ask questions. Start funding research. Prioritizing early investigators is a great starting point. We did that for us. Those grants that we awarded to early career scientists have been the biggest return on investment. And we evaluated that and had a poster at AES, the American Epilepsy Society conference last year. We looked at all the models, both cell and animal, that came from seed funding from us. Also the publications and subsequent funding that stemmed from what we funded. And it was pretty obvious that the early investigators had done a really good job of turning our $50,000 grant into a lot more. Many of them have started their own labs and are working only on SCN2A. If you're established, take stewardship of the research directions, representing the voice of the patient at every level. Serve as a central liaison for researchers. Perform and publish research yourself. Increase funding for research aligned with a strategic roadmap. Organize a conference or a research roundtable. We hired our first chief scientific officer, Sean Egan, as we were coming out of the launched and into the established. So that's a round where we brought in a staff person to help us actually get to the next level. And it's been transformational for us. If you're mature, and as a mature organization, you have the flexibility and the infrastructure to be at the cutting edge of innovation. Dive into the related areas of basic science. Be innovative. Utilize the platform that you've created to amplify discussions and research initiatives effectively. The third pillar is translational research. This involves actively exploring and advancing through the states of discovery, proof of concept, and optimization of therapeutic interventions. The benefits of this are it bridges the gap from discovery, basic science, to clinical application, bench to bedside. It identifies targets, mechanisms of action, and optimizes the therapeutic window. It also creates more shots on goal for novel therapeutics, building and diversifying your pipeline. Examples of ways you can drive the progress in translational research. If you're just beginning, it's kind of hard. I mean, you really, really need scientific infrastructure in place to pursue this, but you'll get there. If you're launched but still figuring things out, infuse the voice of the patient and translational goals into research efforts. Develop a research roadmap. Integrate translational themes into requests for applications. We put out RFAs for specific topics that we want to see improvements on. If you're established, establish an infrastructure to facilitate the translational discussions. Advance your research roadmap. Direct fund translational initiatives. Deploy capital to address the gaps in translation. If you're mature, this is where the ecosystem that you built starts really being leveraged. Be that central liaison in the ecosystem to hear and solve problems between investigators. Stay innovative. Diversify your translational portfolio and act as a liaison between translational assets and biotech industry groups. Okay, we're on the fourth pillar. This is clinical trial research. This is where you are evaluating the efficacy of potential therapeutics and assessing the risk-benefit ratio. These studies are typically performed by biotech and pharma, but there are lots of ways patient organizations can support, and for mature organizations, ways to drive this. The benefits to this pillar, this is the primary pathway to get novel therapeutics to your disease state and community. This research also establishes and reinforces the paths to approval for your condition. Clinical trial research brings added focus, spotlight, and often capital to your disease state. Sorry. Examples of ways that you can work towards driving clinical trial research. Again, if you're just beginning, in order to pursue clinical trial research, you need some scientific infrastructure in place. If you're launched, established, or mature, we think of it as these check boxes. Actually, Sean put together this exact thing for us when he first started, and we started checking these boxes. Is your disease treatable? Is there a proof of concept? Is the therapy commercially viable? Have you defined the market size and the diagnosis rates? You can do this. Are there biomarkers that can support development for accelerated approval? Can treatments be delivered appropriately? Is there a path to approval, a primary endpoint that's been established? Is there enough interest and funding to develop a drug? Is there a willing community to participate in these clinical trials? You can leverage all of the other pillars to check these boxes. And for mature organizations, you have the capacity to do this yourself through a number of different routes, including developing drugs yourself, which is another growing trend. Okay, so I wanted to thank you very much for listening, and if you have any questions, I guess I'm taking those now.
All right, are there any questions in the room or online for Leah?
I can run the microphone to you if you have a question. Hi, I'm Tarn from NINDS. I work in the small business program, and I just was curious, when you said you saw the biggest return on investment from the young investigators, kind of what you were grouping into the young investigator category, and how you were sort of defining return on investment. It sounded like you were just saying they stayed in the field, but I was wondering if there were any other things you looked at.
Thank you. It's a really good question, and I will do my best to answer it, but then I'm also going to give you Sean's email address. Just because he's home with a sick kid doesn't mean he can't answer emails. So we actually had the poster. We emailed every single researcher that we've ever given grants to from 2018 or 19 on, and we asked them to share with us any models that were made from either the funding that we gave them or data that was developed from the funding that we gave them. So they gave us a list of all the models, animal models, a lot of mice models. There's some rats too, and a lot of cell models. So we started to we cataloged those. So now we can share those with other interested researchers and work on getting MTAs, so people don't have to remake those models. We also asked if they got any subsequent funding, and it was really interesting how much funding. I can't quite remember, but I think it was at least sixfold of what we put in. I think, I wish I had put that slide up here, but I'll get the answer to you. It was incredible how much further funding, either FDA or NIH funding, that these investigators were able to get based on the data that was accumulated from our grant. Our grant is called the Action Potential Grant. It's, a little play on words there because we're a sodium channel. And so to qualify for our Action Potential Grant, we have given it to PhD students, but most of them are postdocs that we give these grants to. And what else has come from them? Many of them have started their own lab. They were so inspired by the work that they were doing, coming to our conferences, meeting our kids, that they decided to start their own lab and work directly on SCN2A. So we count that as a big win. But I'm happy to get you that poster. I think it's on our website as well. Of course.
For patient advocacy groups who are in like that just beginning phase, how would you recommend directing monetary resources to sort of, I guess, like promote awareness and start funding research? I know you mentioned funding young investigators, but yeah, I was wondering if you could expand a little bit more on that.
Yeah, that's a great question, and one that I remember very, very clearly. We were just trying to get it on the map. We just wanted anybody to look at it and get and produce some data, get it in the literature, just to get this on the map of something that was interesting, because most researchers were not interested. They actually used to call it Dravet syndrome. Like when my son was diagnosed, they called it Dravet syndrome, and I joined the Dravet group, and that's how I knew it wasn't Dravet. And so in the beginning, we gave out whatever we could pull together, and we didn't really have a systematic way of doing it or vetting research. But we quickly realized that wasn't probably the smartest idea, and so we quickly assembled a scientific advisory board. And we really rely on our SAB. I know some groups put people on their SAB for their reputation. We don't do that. We really use our SAB, our MAB, to advise us, and they help to vet the research and make sure that that it's something that we're going to get something out of. Other ways, it's just awareness, I think, in the beginning too. Getting it out there as much as you can. And remember, you don't have to give $50,000, $100,000 grants. A lot of young researchers or labs are very happy to have $10,000. It helps. Also, another thing that we did that I think was helpful is we co-funded with groups. So we co-funded with American Epilepsy Society. We co-funded with the Autism Science Foundation. And that helped our dollars go further, and I think it helped our reputation as well, to know that we're very collaborative and we work with these other organizations.
All right, thank you. We'll take one more question.
Thank you. For the translational research pillar specifically, I was wondering how you and other organizations are starting to think about intellectual property and partnerships with industry for commercialization. I know some people have taken the venture philanthropy model where it's kind of evergreen and the success can fund. Others are kind of staying silent and not taking intellectual property rights. So I was just curious what your views on that are.
Well, that's the growing trend question right there. That's a really good question, and one that we're still working on figuring out, to be honest. We have tightened up our contracts that for research that we give out. We've had lawyers look at it, and we do ask for a percentage back if something is commercialized. And so I'm happy to share our research contracts with anybody that's interested in looking at it as an example. We've also talked about that for-profit arm of a nonprofit. It's not something that we've done yet, and it's something that's super complicated and risky. But also, I understand why people are doing it, because they're seeing drugs get shelved, and that's devastating to a community when we're waiting and waiting and waiting. It's just. There's also another resource for nonprofit organizations that's free legal advice. I'm going to have to. We the Action. And they work with nonprofit organizations to help with that IP. So if you need a lawyer and you can't afford one, they'll do it for free. They match you. Hopefully that helps.
All right, thank you, Leah. All right, that was a wonderful stage-setting talk, and now we're going to dive more into the last 3 pillars that Leah introduced. And so our next speaker is Dr. Michael Boland. He's the Strategic Director of the Translational and Clinical Research Center for Epilepsy and Neurodevelopmental Disorders, or END, at University of Pennsylvania and the Children's Hospital of Philadelphia. And his talk today is going to be Understanding Your Disease Biology and Physiology.
Thank you, Allison, and thanks to the Patient Advocacy Committee for the opportunity to speak to you today. Leah, that was a terrific stage-setting talk. I'm going to delve into a little bit more of the basic biology of how to understand your disease, and also talk a little bit about clinical research. As Allison mentioned, I'm the strategic director for this new center that bridges both Penn Medicine and Children's Hospital of Philadelphia, called the Center for Epilepsy and Neurodevelopmental Disorders. And we were extremely lucky to receive a $25 million anonymous donation to fund this center. And the goal of the center is to bring gene-targeted therapies to the clinic for STX-BP1 disorder and SYNGAP-related disorder within 5 years. And so we have this huge translational arm of END, as we like to call this, that I won't talk about today. But we're developing ASOs, gene replacement therapies. We're looking at drug repurposing, CRISPR activation therapies. As Leah mentioned, we need a lot of shots on goal, so we're leaving no stone unturned. So I won't talk about any of that work, but I will start by saying that before I was recruited to END about a year ago, I was a assistant professor at Columbia University studying neurodevelopmental disorders. My group collaborated with a number of other groups and studied a number of different neurodevelopmental disorders and pediatric epilepsies, from Fragile X syndrome to GRIN2A, KCNT1, H&RMPU, GNB1, among others. My background is in human pluripotent stem cell models of neurological disease and also genetic mouse models of disease. How do I do that? Okay, no worries. Thank you, I didn't realize that. No, it's perfect. Can you guys help me with that? Yeah, they're working on it. Okay, well, I'm just gonna keep going then. But it's stealing my thunder.
Yeah.
It's totally stealing my thunder right now. So as I was working on neurodevelopmental disorders, 7 years after I started this interest, my first son was born. His name's Lucas, and the day after he was born, I noticed him have several seizures. And I quickly alerted the neonatologist on call, and she agreed that there was something wrong. So he was admitted to the NICU, put on levetiracetam or Keppra, and spent the first week of life in the NICU being poked and prodded. He did EEG, a spinal tap to look for virus in CSF, metabolic panel, MRI, all of which came back negative. We also took blood to send for genetic testing based on my background. I was at the Institute for Genomic Medicine, and I kept thinking the only thing that hasn't come back is the genetic test, and sure enough, Lucas was diagnosed with a frame shift in STX-BP1, which, as many of you know, is automatic pathogenic variant. It's a haploinsufficiency. Lucas is like Ben. Lucas has an amazing smile, and he just touches everyone that you come into contact with. And he works extremely hard in occupational therapy and physical therapy, speech therapy, feeding therapy. He's almost 6 years old. He remains nonverbal, although he's making strides with his AAC device, and his personality is really starting to come through with that, and he's really sassy. He likes to tell his teacher that he's bored with her lessons. So, yeah, they think it's hilarious, but anyway. But he's also non-ambulatory. He just took his first independent steps, 5 of them, about a month ago. So I do think that he's on track to walk sometime in the next year. And I keep telling my wife, be careful what you wish for. Be careful what you wish for. So let me give you a brief background of STXBP1 and STXBP1 encephalopathy, and then very briefly into model development to identify disease-relevant phenotypes, and I'll give examples from mice and from human cortical organoids based on activity differences, neuronal activity differences. And then I want to spend the last 5 minutes or so on clinical research to understand the phenotypic heterogeneity in the patient population. STXBP1, like many of these neurodevelopmental disorders, have a wide spectrum of phenotypic presentation and severity that needs to be understood before you can take any therapy to the clinic. So STXBP1 is a master regulator of synaptic vesicle exocytosis and neurotransmission. It interacts with the snare complex of proteins and regulates how synaptic vesicles are exocytosed. 50% reduction of STXBP1 results in reduced neurotransmission and somewhat conundrum is hyperactivity as a result of reduced neurotransmission, but it's expressed in all neurons, and I think that's a key because it's expressed in both excitatory neurons as well as inhibitory neurons. And with these violin plots, this is single-cell RNA sequencing data from newborn mouse cortex and mouse hippocampus. You can see that the STXBP1 is expressed in all of the neuronal cell types, but not in the non-neuronal cell types such as glia, astrocytes, oligodendrocytes, and microglia, and what have you. So STXBP1 was first identified as a disease gene in 2008. And the presentation for STXBP1 is autosomal dominant inheritance. These are de novo heterozygous mutations. And from everything that we've seen so far in the lab and in the clinic, the primary mechanism of action is loss of function through protein instability. So it has an incidence of about 1 in 33,000, and I said there's a wide clinical phenotypic heterogeneity in that, but almost all kids have epilepsy, particularly within the first year of life. Almost all have some degree of intellectual disability. When this review article came out, we thought that most of the kids have severe to profound intellectual disability, and that has come down a little bit. We know of many more cases now with mild intellectual disability, but most of the kids still do remain with severe cognitive impairment. My son Lucas has an IQ of about 30, and he's almost 6 years old, and in many ways he's in some ways an infant, in others a young toddler. And almost all of the kids, because of this cognitive impairment, have motor planning difficulty, so they have motor features. There are a number of different epileptic syndromes associated with mutations in STXBP1, but the primary presentation is this early onset epileptic encephalopathy. So whenever you're thinking about developing disease models for identifying clinically relevant phenotypes or disease-relevant phenotypes on a basic level, you have to realize that none of these models are that good. They all have their pros and cons. So it's really important to develop a number of different models. With my background in mouse models and human pluripotent stem cells, at Columbia, we immediately started working on developing both of genetic mouse models and human-induced pluripotent stem cells that we could then coax into neuronal networks or into cortical organoids to study activity differences. And we take the same philosophy at the End Center at Penn. So there are a number of different ways that you can look at different levels of complexity in these different disease models. You can look at EEG or behavioral characteristics in the whole animal, in the mouse. You can look at brain anatomical differences in the mouse. And then at a little bit of a lower level, you're looking at neuronal networks, whether these are derived from human-induced pluripotent stem cells or if they're primary networks pulled from the mouse brain. You can use multi-electrode arrays to study network activity differences. You can look at transcriptomic differences. And similarly, with single cells, you can use very high-resolution patch clamp electrophysiology as well as single-cell transcriptomics. I have data on every one of these modalities that I'm not going to share with you today. I'll focus on neurophysiology because of the mechanism of action of STXBP1 and its relationship to how neurons communicate with each other. So we have a number of different. What is this? Here we go. So we've created, this is at the Center for Epilepsy and Neurodevelopmental Disorders. These are the primary models that we're working with. We have the traditional heterozygous mouse knockout, STXBP1, and we've also made a humanized mouse model where we've replaced the mouse locus of STXBP1 with the human locus of STXBP1, and this includes all of the upstream and downstream regulatory elements because many of the therapies that we're developing are human-specific, whether they be ASOs or CRISPR-A or something like that. So we need to be able to test them in vivo in an animal model, but we need that animal model to have the human sequences that we need. And so we've developed the humanized mouse locus, and we're also starting to develop certain patient-specific variants on the humanized background. For instance, this arginine 367 to premature termination codon, among other missense variants. For human-induced pluripotent stem cells, they kind of come in many different flavors. They're all as different as the people that they're derived from, so you have to be careful from a standpoint of controls. Which controls are you going to use, and how rigorous are you going to be with validating the results related to your controls? And so two of the popular models are this trio familial model where you have iPS cells derived from mom and dad as well as the proband. We have those, as well as isogenic models where you can derive iPS cells from the patient, him or herself, and then use CRISPR-Cas9 to correct the mutation, generating the isogenic control. Or on the flip side, you can take an unaffected iPS cell line and use CRISPR-Cas9 to knock in different variants into that background. This gives you a lot of power because you can generate an allelic series to test different therapeutic modalities on a uniform genetic background. And it also gives you a really good way to define your disease mechanism, which is absolutely crucial for taking any therapy to the clinic. You definitely don't want to take something that upregulates expression of a disease gene if there's a dominant negative phenotype. You're going to make things worse. You want to be able to tailor your therapeutic modalities for your disease mechanism. Luckily for STXBP1, the primary mechanism is loss of function. This clicker is really slow. All right. So a little bit of data. I said I would focus on neurophysiological phenotypes. So in the STXBP1 heterozygous knockout mouse, if you perform EEG, and this has been done in a number of labs, this is actually a really robust phenotype. They have these spike and wave discharges, which are similar to absence seizures in humans. And this is a very characteristic phenotype of STXBP1 mouse models. You've probably may have heard other talks like Mingshan Xue and Charles Chen talked yesterday about some of their work with STXBP1 mouse models. You can also use, in an ex vivo form, means to develop investigate brain-specific activity patterns. And so in this case, what you would do is you would take the neurons from the brain of a newborn mouse and you plate them onto these multi-electrode array plates. So you plate neurons in each of these wells. Each of these wells contains 16 electrodes that measure the local field potentials from neuronal activity. And so you can start to put together activity differences between different genotypes. And what I want you to appreciate here are what's in A versus what's in B, which is quantification for the different representative raster plots that are shown in A. But you can see that in the cortex and in the hippocampus, you have very different activity patterns in STXBP1 versus wild type. You're looking at the 16 electrodes from 1 to 16 on the y-axis and time on the x-axis. And so what happens is you get these really large bursts that have a lot of time in between the bursts in STXBP1, cortex versus wild type mice. And that will become important in just a minute. But these are really highly reproducible phenotypes that you can start to build relevant platforms for testing therapeutic efficacy. We also, my background, I had to make stem cells for my son, of course, and confirm in neurons derived from those stem cells that yes, it is true haploinsufficiency as we expected. There is a more than 50% reduction of protein. And then we generated human cortical organoids from those stem cells to look at activity patterns in intact organoids by calcium imaging. And this is an example of one of those organoids that's almost 4 months old. They're about 3 millimeters in diameter. And if I play this movie, if it plays, what you'll appreciate are this really large bursting of synchronized activity across all of the neurons in the organoid that occurs at a somewhat regular fashion. And then there are these little blips of microcircuit activity that we think might be related to essentially seizure activity in the organoid. And if we quantify this, you can see hopefully you can see that the STXBP1, it looks very different from the controls. Each of these lines is a different region of interest where we've quantified the neuronal activity. And you can see that the controls have a very, very synchronized periodic periodicity. And the STXBP1 have these long interburst intervals, a larger, wider burst duration, which includes a higher number of spikes in each burst. And I want you to remember that that's very similar to what we saw in multi-electrode array from our mouse model, indicating that this might actually be a cross-species neural activity phenotype that we can really rely on to test therapeutic efficacy. So with that, I want to shift gears a little bit in the remaining minutes, talk about clinical research to understand the patient population. And I just want to touch on 3 different areas: disease concept models, retrospective and prospective natural history studies. So in 2021, The STXBP1 Foundation polled a number of caregivers of STXBP1 individuals about what were the most stressful symptoms the caregivers dealt with. And not surprisingly, seizures were high on the list, but speech and communication and cognition are also high and even rank higher than seizures. It's very difficult to not be able to communicate with your child, or have a child that's 5 or 6 years old that's not potty trained and can't dress themselves. So another way to look at this is through the idea of a disease concept model. And this is a more robust caregiver survey where you interview a number of different caregivers and identify different symptoms and aspects of life from a medical standpoint and how those symptoms impact daily life. And so here I'm showing you results from the disease concept model for STXBP1, and you can see that on the bottom you have age, and then on the X or on the Y-axis you have the density or the proportion of impact or symptoms. And tone and seizures and gross motor are very prevalent very early, and I told you that STXBP1 is largely characterized by very early seizures in the first year of life. Another way to look at this with a little more data-driven approach is through retrospective natural history study through claims and EHR mining. And we've done this for 162 STXBP1 patients and have defined both developmental outcomes, such as what are shown here. You can see that, so the dashed colored lines is the age at which 75% of typically developing kids reach that milestone. And you can see that, for instance, the furthest right shifted are the ability to initiate words and the ability to walk. Lucas is a perfect example of this. He fits this trend perfectly. He's almost 6 years old, but he still can't talk or walk. We can also use these data mining approaches to more accurately define the seizure landscape for STXBP1. And as I mentioned, STXBP1 is a disorder characterized by really severe seizures in the first year of life, and you can appreciate that here with the infantile spasms and focal onset seizures that you can see in the first year. And in the inset, you can look at the severity of seizures in this cohort of kids. Running through this data, we can perform virtual clinical trials and to look for age-related epochs where the seizure frequency is stable enough where we could see a 20% reduction in seizures from any therapeutic agent. And so you can see that after compiling the seizure landscape, we can actually see two areas of age from about 1 year of life to 5 years, and then from about 7 years to 12 years old, where the seizure presentation and frequency and heterogeneity is stable enough where we could potentially use these age ranges in clinical trials with seizure outcome as a primary measurement. And in the last couple of minutes, I'll want to talk about our really large multi-site, prospective multi-site natural history study now. This is, we call it the STAR study, and the goal is to look at 100 kids in the first year. We started this in July of last year, and we'll meet that goal by July of this year. We've already seen through middle of June, we will have seen 75 STXBP1 kids in the clinic. And the study is designed to capture a wide breadth of phenotypes for STXBP1. We want to capture cognition. We want to capture gross motor. We want to capture EEG, So a typical day is the kids are seen by a developmental pediatrician, a physical therapist, an occupational therapist, a neurologist. We do developmental testing, and before that, we send out questionnaires and look for patient-reported outcomes that we can then essentially compile the patient-reported outcomes with the clinical data generated at the natural history study. So that's the first year. And then we start to do a longitudinal phase where we start to look at the kids every 6 months for a period of 2 to 3 years. And in this way, we can define how the phenotype expands or what it looks like on an individual basis and in a larger cohort over time. The idea here is to try and use this data as an external control arm or in lieu of placebo in a clinical trial. One more example of a potential biomarker that's age-dependent is this idea of using EEG and different measurements from EEG. It's such a rich data set. We're just now starting to appreciate its roles in everything from normal cognition outside of seizure tracking. And so if we look at the power spectral density, which is essentially a distribution of signal power versus frequency, you can start to identify intellectual disability that separates from control individuals with or without ID. And also, we can start to develop disease-specific signatures. For instance, in infants, we can now identify STXBP1 distinctly from other neurodevelopmental disorders, whereas that distinction becomes less relevant or not measurable in older children. So there's an age dependence here, just like what we saw with the seizures. So with that, I want to finish with this slide that encompasses what we at END think is really important. And hopefully both the translational research and your clinical research are driven by the basic research and the clinical phenotyping that Leah so eloquently described. And these should work in parallel. Ideally, these work in parallel. So many times groups get so caught up in translational research they forget to define their patient population. And so they have this beautiful ASO, and they don't know who to treat with it or what age ranges are the best appropriate age or which mutations might be the best or most efficacious. So it really, both of these have to, are important to develop in parallel. And the clinical research, I think, is often more difficult because you're dealing with patients rather than mice that you can just breed up or cell lines or whatnot. So it's really important to start this early. With that, I'll thank the large team of very dedicated individuals at END. You see Lucas and I in the front, and the director of our center, Ben Prosser. His daughter Lucy also has STXBP1, and so we have a really close motivation for STXBP1, but also for SYNGAP as well. And then I'll also thank a few people from Columbia that helped with the model characterization that I showed earlier. Thank you.
Thank you for that beautiful talk. We'll just take maybe 1 or 2 questions.
I put everyone to sleep.
It's just so clearly stated. Yes?
It's a great question. And so so many of the trials, like the first trial in STXBP1 was 4-phenylbutyrate. and the primary endpoint was seizures. And that trial really didn't go all that well. And I worry, Allison and I were just talking about this yesterday, I worry that upcoming trials will only focus on seizures. And that's why we're trying to develop outcome measures in a number of different areas. I think for STX-BP1, one of the best ones is motor development, because that relates to cognitive motor planning. And almost all of these kids have delay in motor development. Some will walk by the time they're 2 or 2.5, but most don't. Most don't walk until they're at least 4 or 5 years old. And so I think that if you can start to, what we really want to do is start to define age-appropriate outcome measures, which will be difficult, but I think given the fact that we're looking at cognition, motor, fine motor, gross motor, fine motor, we're looking at this wide heterogeneity of potential outcome measures. We'll start to define disease-specific outcome measures. So what we see for STX-BP1 is certainly not what we're going to use for SYNGAP. For SYNGAP, I want to look at sleep and behavior as more of an endpoint than motor deficits, because they're not very affected by motor deficits. So I think it's going to come down to disease-relevant, age and activity-specific outcome measures.
Thank you, Dr. Boland.
All right, moving right along. So this takes us into our translational pillar. So we're really happy to welcome Dr. Joni Rutter. She's the director of the National Center for Advancing Translational Sciences, or NCATS, and her talk is going to be Rare Disease Research Innovations.
Thank you so much. I really appreciate it. Everything okay? Okay, good. All right, so I've learned that there's a delay, and hopefully it's just the right-hand button, and then I'll be set here. Thank you so much to the organizers, to the panelists who are with me today. It's a great honor to be here to talk with you a little bit about what NCATS, the National Center for Advancing Translational Sciences, is doing. So we're primarily about that third pillar that Leah talked about, translational research. And what translational science is a really learning about all the translational research that's being conducted, understanding where the bottlenecks are, and innovating on those bottlenecks so that we can go faster, better, more effective and efficient in that translational science pipeline. And so what I'll talk a little bit about today is really how we're thinking about re-engineering that translational pipeline. Our public health challenge at NCATS is essentially this pipeline. And a big area of unmet need is in the space of rare diseases. So that's really where we focus. But there are some big bottlenecks in doing the research along the way. So that's what I largely want to talk a little bit about today, is how we think about the research in this space, how we think about tackling the bottlenecks in this pipeline. Because you know that if you have a pipe, if there are clogs in that pipe, the flow of anything going through that pipe is dictated by the clogs. So we want to unclog the pipe as much as possible to get more treatments than really through that pipeline, if you follow that analogy. And this is important because there are over 10,000 rare diseases, as many of you know in this room, let alone all diseases. And only 5% of those diseases have therapies. And so we've heard about a couple of diseases so far today, and there are many more that you'll hear about over the course of this meeting. But the important pieces in understanding about the bottlenecks in here is that it can take about 10 to 15 years to get a candidate to the point of testing that candidate in humans. And then another decade or so before it actually gets into medicine cabinets, so to speak. And 90% of the time, that whole pipeline fails. So they're mainly because of these bottlenecks. And so there's a real need to understand where these bottlenecks are and how to address them so that we can decrease the clogs in the pipe and things can go better and faster. So our mission essentially, Oh, it didn't, let's see. There we go. Oh. No. It's going the wrong way now. Like this. Okay, there we go. There we go, that's the one. Okay. So essentially we, at NCATS, we have four, I would call it fundamental de-risking approaches for the work that we do in tackling the clogs in this pipeline. We want to first address diseases in a more than one disease at a time fashion. Given that there are 10,000 diseases, we want to ensure that we have ways in which we can enable people to go more than one disease at a time. So a lot of what we build are platform-based approaches that can enable this work. We want to also consider how to develop better predictive models, a lot of like what Michael just talked about in terms of those organoids and other kinds of tissue chip models and other human-based cell models that we can use to do this. And then we also want to think about how clinical trials can be more impactful and more inclusive, ensuring that the work we do in clinical trials is as generalizable as possible for the patients who need them. And to do this, we need to ensure that we work with patients and patient advocacy groups, because that's going to be central to the work that we do in this theme. And then lastly, we want to leverage real-world data, big data, data science to address the public health needs that we know exist. And so I'll talk a little bit about some of these aspects over the course of this presentation. So our mission essentially is to address these barriers by turning scientific findings into health solutions. And our goals are, we have three major sort of audacious goals that we are using to address these. One is to bring more treatments, and by that, we want to change that number of 5% that I mentioned earlier. There are 5% of therapies for diseases. I want to change that number to 25%. That's a big leap. But I think with the tools and technologies that we have today that we're hearing about at this conference, for example, among those, we have an opportunity to really make headway here. And I would rather fail at 25% than succeed at 6%. So we want to set that bar high. In addition, we want to make sure that what we do, we bring more treatments for all people. We want to dramatically increase inclusivity in every area that we support, and that's critically important to the work that we do. And then finally, more quickly. So by building platform-based approaches, we think that we can enable diagnostics and therapeutics to reach people twice as fast, at least. And we know that in the rare disease space, this is critically important. Many times rare diseases are diagnosed, but it takes, on average, about 7 years for that diagnosis to occur. That 7-year window is a critical window. It is far too long right now. And we're missing that critical stage where perhaps we might be able to get some modifying, disease-modifying treatments to those patients earlier and faster. So that's a really critical reason why this last goal is so critical and so important. So with these goals, in a sense, we are focused on de-risking the translational science pipeline. And sometimes I call it more distributing that risk. We are de-risking it, but we're not taking that risk down to zero. Things that we build and enable, ultimately to get them to commercial implementation and testing in clinical trials, they can still fail. But we can distribute that risk a little bit more by doing as much as we can upfront and early on and enabling these technologies to build and enable more people to access those kinds of therapeutics faster. So at NCATS, we fund research, we do research, and we partner to enable all of these to happen. And we think of ourselves as the dot connectors. So we work with .coms, .govs, .edus, and .orgs, and patient advocacy group organizations are a big part of that. So one of our key flagship programs here is the Rare Disease Clinical Research Network. And this is a network of about 20 different organizations across the country that is funded by all of the folks here shown on the bottom of this slide. Across the NIH, we have about 10 different institute and center partners that help us with this, and NCATS is the steward of this program. We have 20 groups, as I mentioned, across the country that focus on a variety of rare disease groups or rare diseases. And over the course of the work that gets conducted within each of these networks, they have to at least study 3 different rare diseases within those groups. That's the requirement. However, most of them study many more, up to 20 diseases or even a little bit more than that. So right now, as a collective across those 20 different networks, we are studying over 280 rare diseases within these groups. And most importantly, we have a coalition of patient advocacy groups who are involved in each of these research networks as a part of the science that gets conducted. There's a coalition of these patient advocacy groups as well. They have a steering committee that helps activities across the entire consortium of the Rare Disease Clinical Research Network. And I think that is the most disruptive activity for the Rare Disease Clinical Research Network. And I like to say that this is one of the more innovative programs that we support. It's not just about the infrastructure for supporting rare diseases, but because of all of the work that they've been able to do over the last 20 years, they have enabled 12 new drug approvals to date. And if you remember when I said that it takes 10 to 15 years to get one drug to approval, that's a pretty remarkable number for this collective of an organization, and we look forward to hearing about more new drug approvals as well. So this is really one of those engines that really keeps the research space going in the rare disease innovation space. So a little bit more about the Rare Disease Clinical Research Network sites. As I mentioned, they are all across the country, and they are even international. This international component is critically important. As you know, individuals with rare diseases are rare in many cases, but collectively they can be much more common. And so it is important to work across the globe when we're thinking about rare diseases so that we can get as many patients who are available for the research that we're supporting. So we've, over the years, actually increased our numbers in terms of the sites that we're supporting, and also across the globe, the number of sites that we're supporting. And I think that's been really effective in helping us get to that high number of new drug approvals as well. In addition, we've also, I think because of the collective of this network as well, there is an incredible proliferation of publications, collaborations, and co-authorships across this network. And again, that breeds this innovation, and it breeds more, pushing more towards therapeutics. And so with the numbers of organizations that have increased, you can see that by adding even our international collaborations as well, we've dramatically increased the number of publications that we've been able to reach. And these are also just shown in another slides in terms of how these authorships are relayed and the numbers of networks that we're able to help enable and expand on the work that we're doing. And then lastly, one of the things that I wanted to mention about the RDCRN group is that we are building quite a large data network or a data environment, and hopefully it will advance in just a second. It may not advance. There we go. We're building a large data environment as well. So each of those 20 different networks with their nodes are contributing data to a centralized ecosystem of information on all of those rare diseases. Right now, it is essentially an operational environment where we have the consented data, we have de-identified data, and we have also metadata of understanding how to do quality control within those data, understanding how those data need to be used based on the consent, et cetera. And then eventually, we're going to expand this in our next iteration of the Rare Disease Clinical Research Network to a more shared environment where all of the networks can contribute data, and then over time, they will be ultimately made publicly available for others to use as a public-based resource. And there will be a data access committee for us to be able to manage and control how these data will be accessed and used. So we're looking forward to this environment. It's in the process of being built, but please stay tuned because I think this will be a very rich resource for the rare disease community. So essentially with the Rare Disease Clinical Research Network, we're taking a lot of the activities, the infrastructure that's built across the country, all of the different over 280 rare diseases that are being studied, and creating essentially a research community that can pull together and enable a lot more of this sort of pipeline building, bottleneck busting activities to occur for translational science to proceed even faster. And then lastly, for the RDCRN, I want to mention that we've just opened up a new funding opportunity for the Rare Disease Clinical Research Network. And as the patient advocacy group organizations, which I imagine many of you are part of different patient advocacy groups, if you're interested or working with scientists who may be interested, I wanted to make sure that you're aware of this and can work on perhaps a solicitation that is due August 13th. So you still have a couple months to go. And we hope that we see a variety of applications during this round of opportunity for the RDCRN. We look forward to bringing in new activities within the rare disease space as well. So that's one of the major workhorses within NCATS and our rare disease space. I now want to just talk about some other resources and activities that we have that may also be helpful for you as well. Oh, I did want to mention too that within this RFA, it says very explicitly that the lived perspective of the patients and their needs, their priorities, must be meaningfully incorporated into decisions and activities of the proposed RDCRC, with related patient advocacy groups having an active partnership role within the RCDRC. So I wanted to just make sure that that was very much highlighted. This is front and center in the purpose of the whole Rare Disease Research Network. And so this is critically important to the work that we do in a way of funding some of the rare disease advocacy work that you all do. Now, there are some other opportunities as well, and I'll get into those. So the other resources that we have are highlighted here. At NCATS, we've over the last several years have created a variety of tools and resources. The first is the Genetics and Rare Disease Information Center. This is one of the most popular websites at the NIH overall, let alone at NCATS. But essentially, it's one of the first places that people go if their child is diagnosed or their family member is diagnosed with a rare disease. This is a place where you can go for all sorts of information about that particular rare disease diagnosis, or maybe you don't even have a diagnosis yet and you don't know what to do. There's a call center associated with the GARD website. So please take a look at the GARD for all sorts of information that would be available. It can tell you how to find researchers, how to find patient advocacy groups, how to find clinical trials, et cetera, if any of those things are known about the disease of interest. The second one I'll mention is the toolkit, and I'm going to actually come back to this one a little bit more, so I won't spend too much time on it, but it's essentially a toolkit for patient groups that will enable your ability to find researchers within the community who may be working on a particular rare disease that you're interested in, and it gives you some tools and resources to be able to do that, especially as you're thinking about therapeutic development. A lot of what Michael described for the disease that he talked about is exactly sort of the pathways and sort of stepwise approaches that this toolkit provides as well in a structured way. And then lastly, RADAR is a rare disease registry program, and this is a place where we collect patient registries so that we can help people find those and use those as they need to. So back to the toolkit for sort of the patient-focused therapy development. This is a platform approach that we've used to really help patient groups be more participatory in the therapeutic development of their specific disease that they're interested in. This toolkit helps explain how the research process works, and it illustrates a variety of ways in which groups can collaborate, network, especially amongst themselves, but also with the researchers out there who are doing specific activities. And it provides links to those other resources that are made by and for the patient community. So some of those links and QR codes are shown down below for the toolkit itself, but also for sharing resources that patient advocacy groups have generated. And this is a way of sort of bringing those together to help others who may be newer to the rare disease space to help them out in understanding what your process has been. Now, one sort of way in which you can go to the site, if you actually click on the toolkit on the left-hand side here, you land on a page that looks something like this with these 4 cards in front of you. And so this is essentially a toolkit from discovery. This is sort of like the basic science tier. the prepare for clinical trials, that's sort of the clinical tier. The clinical trials and FDA review, this is help in understanding FDA and regulatory requirements that are needed. And then what happens after FDA approval. So let's just take a quick look at what happens if you click on one of these sites. We're going to click on the prepare for clinical trials tier. So when you click on that, it opens up to a new page that tells you a little bit more about how you can prepare for your clinical trials. And it gives you, again, another set of cards that you can look through. And so you can establish the clinical sites, help industry with clinical trial design, et cetera. And then if you click on those cards, they turn over. So that's where the red arrow is. So I clicked on one of those cards so that you can find out ways to ensure that you're invited by industry sponsors to meetings with the FDA. And it tells you how to get those connections made so that your voices are heard with the FDA as well. And the FDA holds a variety of patient advocacy group meetings. And so I want to make sure that you're aware of how to access those and get plugged into those sites and resources. So that's the toolkit for patient-focused therapy development, for example. Another activity that we support at NCATS is conference grants. And Leah mentioned that one of the activities that's really important for new patient advocacy groups that are starting out or are somewhat mature is actually thinking about holding conferences or workshops. And NCAT supports scientific meetings. These conferences and their workshops, they're called an R13 mechanism. And we support a lot of these in rare diseases. And often it doesn't take a lot of money to support these activities, but this is a way that we can help make sure that there's a mechanism in place for you to apply for and get at least access to submitting an application for this. These are peer-reviewed and scored, and so we're held to the confines of that scoring process. But we do seek applications for conferences that include participation of relevant patient support groups in a variety of these sorts of meetings. We want to make sure that we're leveraging the most advanced breakthroughs in what that conference is going to look to support, and then helping you set some of those clinical practices, thinking about natural history studies, for example, or thinking about clinical guidelines. So again, these are small amounts of money for conferences, from $3,000 to $25,000, but this is another way in which we can support those sorts of activities. And there's a link here below. I'm sorry I didn't put the QR code. I forgot, but there's a link here in case you're interested. And then one other note of mention in terms of the natural history studies that have been mentioned throughout. These are incredibly important for patient advocacy groups to be considering and how they develop these natural history studies and the repositories for the data that are collected in them. And the Reagan-Udall Foundation, along with us at NCATS and our rare disease groups, the FDA and CEDAR, are hosting a rare disease natural history studies meeting, and it's geared towards patient advocacy groups and clinical researchers. It will address the role and design of these sorts of registries and natural history studies. It will really focus on how to collect data that are fit for regulatory purposes and the use of registries and natural history studies to inform regulatory decisions, some of which were discussed earlier in this panel presentation. So I hope that you take a look at this. This is happening, I think it's next week, right? May 13 is next week, so happening soon. All right, so I'm going to end really on, well, actually, I want to mention this first. This is another area where there's an opportunity for rare disease communities to get involved in raising awareness about rare diseases writ large. And NCATS hosted a challenge in 2020, and after that, the pandemic hit. We kind of paused this, but we hope that this will continue to move forward. So stay tuned on these challenges, but they've been enormously successful in raising awareness of rare diseases. And so I put the QR code here for you to take a look at some of the past challenge winners, how it's really helped raise awareness of them. And this one is a Friedreich's ataxia patient, Jacob Thompson, who did a spoken word message. It's really inspiring, and I encourage you to take a look at it. All right, I'm going to switch gears to just some of the innovations that I wanted to mention that NCATS is also working on for the rare disease space. So 2 major themes I want to talk about here. One is predictive modeling that Michael also mentioned as well, so I won't spend too much time on this. But one of the key things about rare diseases, with over 10,000 rare diseases, it's very difficult to build animal models. It's challenging, it's hard, and not all rare diseases are going to have the ability to do so. So we want to be thinking about how we can effectively create models for rare diseases that are not using animal models. And so this is one of those areas where we're making predictive models to be able to do that. So a lot of this is about creating 3D organ systems, 3D tissue chip-based models. And as you increase the physiological complexity of these models, you actually decrease the high throughput capability within this space. And this is one of those areas that NCATS is also working on. As we start to build the complexity of these models, we want to also try to maintain as much of the high throughput complexity that we can. I don't think that this video is gonna show, but this is basically a robotic arm that shows you how we automate all of the cell culture that we do for the induced pluripotent stem cells and generating that. Automation is always, it hands down beats manual manipulation of cell culture 100% of the time, no matter what variable you look at. So this is a really critical workhorse for us to develop these types of models. And then when we develop these models, we can take blood cells from patients with rare diseases, for example. We can create iPS cells, much like Michael talked about earlier, and we can differentiate them into just about any type of end sort of cell type that we need to then build these tissues on chips or 3D bioprinted kinds of organ systems or multiple organs on a chip. And this is turning out to be very fruitful. We have a couple of programs in this space, and there is now an NIH Common Fund program that will be looking at the use of these types of in vitro models along with in silico models and in chemical models to really help ramp up and define how we can move these through the FDA pipeline. So these will be really critical, and I encourage you to keep looking at this space and how it might be useful to you. And then finally, we've been involved in a rare disease workshop that is talking about how we use these models. And one of the key features that we've been thinking about is in precision medicine, of course, and in gene therapy. And so NCATS is supporting a variety of gene therapy approaches for a couple of different rare diseases. And we're using these models to test exactly those AAV therapies in this space. This particular model is looking at congenital myasthenic syndrome. So this is a neuromuscular junction. And so we've taken iPS cells from a patient with this particular disease. And when we infuse the AAV virus for the gene therapy, we see return of the muscle twitching. And so that gives us that functional readout. So this is a very new tool that we're able to use in our disposal. And then I'm going to talk a little bit more about sort of how we use real-world data in understanding rare diseases as well. And I'm just going to end on this. We have a variety of activities in understanding how we strategize around drug repurposing, because if we can do drug repurposing, that means that there's a drug already in existence that may be through phase 1 clinical trials, so may have safety data with it, but doesn't necessarily have an indication. So this is one way in which we can try to identify some indications for those existing therapeutics. If we do that, it takes a lot of years off the life of us developing then that compound for therapeutic use. But one of the key features in how we've used this is in our data systems. And so we have a program called the NCATS Biomedical Data Translator. This is essentially a group of over 150 resources of publicly available information, of genetic information, protein information that has been amassed over many, many years, decades even. And we've put that into this sort of AI-based program to generate knowledge based on queries that we can ask the system. And so one query that came to us was one on Shine syndrome. A physician had a rare disease patient with this disease called Shine syndrome, sleep disorders, hypotonia, intellectual disabilities, neurological disorders, and epilepsy. That's where Shine syndrome comes from. And so using Translator, you simply input into the query, what is a drug that may up or down regulate my gene of interest? And SHINE is caused by a haploinsufficiency of DLG4 gene. And so we asked translator this question: what can upregulate DLG4? And it turns out there were a variety of things that came back in translator, but one of those things that came back was something called guanfacine, that guanfacine may increase DLG4. And because guanfacine is a known drug, it's commonly used, especially for, it's a common blood pressure medication. So the physician tried this on the patient because it was a relatively safe drug and we knew a lot about it. And it turns out that 5 months, well, actually, the mothers had sent in some coloring diagrams from her little girl with SHINE syndrome. You can see it's kind of erratic. It's not really within the lines. But at 5 months after post-treatment of guanfacine, the mother sent in this picture and said, Peppa Pig has never looked so good. So this is another possible way, number one, of looking at the phenotypic characterization of these types of diseases by looking at coloring and motor skills, but also just really looking at improvement of these types of medications. And so with that, I will stop there and thank you for your attention.
Thank you so much, Dr. Rutter. That was a really wonderful overview of all the opportunities at CATS. Unfortunately, we are running a little over on time, so I would like to ask the audience if you have any questions for Dr. Rutter. Hopefully you can find her around the conference, but we'll need to move on to our next speaker. Thank you so much. Yeah, and so our next speaker is going to be Dr. Richard Finkel, and he is coming to us from St. Jude's Children's Research Hospital, and he's going to talk us through clinical trials in rare disease.
Thank you to the organizers for the opportunity to come and participate, and thanks to Leah, Michael, and Joni for setting the stage. And I don't have a whole lot to add, but I'll try to give a few thoughts. As a clinician, I see patients. Everyone that I see is pretty much a child with a genetic disease. It's a rare disease, and we try to focus on how to help these children and their families. I'm going to try to address some topics directed to patient advocacy groups. These are my disclosures. Just to say, I actually made these slides without the use of AI. It may be the last time I do this, but here we go. Okay. And, okay, we'll just pause a moment here. Okay, so what do we mean by rare and ultra-rare diseases? So a few definitions. So it varies. In the U.S., the FDA has a definition of less than 200,000 individuals in the U.S. living with that disease. But in Europe, it varies. It could be less than 1 in 2,000 individuals living with that disease, so it varies. But we're going to use the U.S. definition. Now, there's also something called ultra-rare, and there's no formal definition for this, but generally speaking, it's less than 1 in 50,000 individuals living with this, so a good bit different from the rare. And then more recently, maybe not so recently, there's some terms called hyper-rare or nano-rare, and this has to do with personalized medicine, where drugs are being developed for a single individual with a particular mutation or genetic variant. Okay, and that may represent just 1 in 100 million, or 3 in the United States, or maybe even just a single individual. So there's a spectrum here, is what I'm saying. Okay, where do you point this thing? Okay. Okay, so you heard about 10,000. Well, this slide was made a couple years ago, so it's gone from 7,000 rare diseases now. It's felt there's 10,000. There are probably more. But the important thing is that on average, there are about 4,000 patients. That's the median, okay? And there might be 1, and there might be 200,000. But the median is about 4,000. So these are uncommon, but still common enough for pharmaceutical companies to take an interest in it and develop a drug and get a return on their investment. But if you do the math of 7,000 times 4,000, you get 28 million. So it's about 1 in 10 individuals in the United States are living with a rare disease. And it's important to identify that 2/3 of these are children, 30% die before they reach their 5th birthday, so there's high unmet need. These are catastrophic diseases in many cases. And importantly, about 80% of these are genetically based. And as you heard earlier, only about 5% have an FDA-approved drug for that condition. And that's not so unusual. In pediatrics, most of the drugs we use are not FDA-approved for children, so we're using them off-label. But here there's a chance to develop something that's targeted. So quickly, and this has already been gone through earlier, so I'm just going to skip over some of the clinical trial design topics, but also how does it relate to the patient participation and the patient advocacy group? So you've heard from the earlier speakers about drug discovery, testing on cell lines, animals, but from the patient point of view, you want to make sure you're capturing what is clinically meaningful, and you heard a lot of that from Michael, the importance of having skin biopsies and setting up cell lines. You can't force anyone to do it, but if you can emphasize the importance to set up cell lines, I think, and how that promotes some of the preclinical development, that I think most parents will agree to that. And then also there are different types of animal models, and some are better than others, and that none of them are perfect. So sometimes you need several animal models to identify different aspects of the disease. I'm running out of juice here. There we go. So clinical trial readiness, and again, you've heard about this, the importance of identifying natural history studies. So you need rigorous natural history studies, and Michael pointed this out quite well in his. So on one hand, you want to identify the full spectrum of patients with that disease. On the other hand, you want to identify what would be a homogeneous group that you would use in a clinical trial. So there's different objectives here. You want to identify outcome measures. You want to get patient samples, such as blood or other biopsy tissue. But also, depending on the disease, you may want to get MRI scans or EEGs. And these may or may not be paid for by insurance. If it's part of standard of care, yes. If it not, you need some resources to be able to identify these different biomarkers. So you've heard about this already, the preclinical research side of things, so I'm not going to repeat that. But I do want to talk about, just briefly, where can patient advocacy groups come in to support clinical trial readiness? So the support for natural history studies, things that you've heard from others. But on the next slide, maybe. Okay, so I think there's an expanding role for patient advocacy groups. Part of it is to provide necessary messaging, and you can serve as an important neutral party to get the word out responsibly. There's a lot on social media, and those are all true, but it's based on the personal experience of maybe a single patient or parent. It doesn't necessarily represent the disease at large. You can also provide the patient voice. That can be one-on-one discussions privately. It can be surveys. It can be through different groups that where you're trying to generate information to support the clinical trial development, as Michael pointed out. And importantly, to identify what is clinically meaningful and how do you rank order that. So maybe it's a seizure disorder, but maybe seizures aren't the primary thing that affects the child or the parent. So that, I think, was a really important point that you heard earlier. You can sponsor meetings and workshops, as you've heard, and you can engage in each of these, importantly at the NIH in their workshops with the FDA. Get yourself invited to present the patient voice. And you heard earlier about generating revenue for sustainability of your group. And that's becoming increasingly common from my observation, but it also sometimes creates a problem when you have more than one patient advocacy group for a condition because they get into a competitive mode because they're generating data which is important to pharma, but they're not really sharing it, and they're not aligning the data, so there's actually, it can create some confusion. So you're familiar with the traditional clinical trial pathway, which gets modified when we talk about rare diseases. But I just want to point out on this second line here that, and this is data coming from the FDA, out of 1,000 clinical trials that start with phase 1, only about 70%, and that's the dose finding and safety. So 70% get to the phase 2, which is safety proof of concept, and a third of those get to phase 3, which is the big proof of efficacy and safety. And if you do the math, really only about 6% of studies that start with phase 1 actually get their package together and submitted to the FDA. And not all 6% of those get approved. Okay, so you want to increase your odds, right? You want to de-risk this. So you who work in patient advocacy groups really have an important role at each of these points, is the point I'm trying to make, to try to de-risk it and increase the success rate. So the challenges in rare diseases, okay, you have fewer patients. You cannot conduct big randomized clinical trials with 300 patients in each arm. That just isn't feasible. And I'm going to just talk while I'm trying to press buttons here. The FDA typically wants you to start a drug trial in adults, but the disease may be more relevant in children. And the intervention in children, you might actually be able to identify a response. So sometimes there's a conundrum where you have to start in adults because adults can give informed consent, but they may not be the optimal group to study. Conversely, children are often not studied in big trials because there's nothing forcing pharma to do it, even though there's some goodies that they can get from the FDA. But even when drugs are approved for adults, we don't have information to understand the safety, the metabolism, the dosing in children. So as a patient advocacy group, you want to make sure, and I'm sort of waving a banner here, but that if a study is going to be done in adults and the disease exists in children, which is almost always the case, try to urge and prompt and make sure that children are being included as a cohort at some point. Maybe not initially, but make sure that they're being included. Now, at a practical level, these patients can be fragile. So if you have rare diseases, you're not going to have multiple sites. You're not going to have 30 sites in the United States. You might have 4. But getting the patients to travel long distances can be a challenge, particularly if they're fragile. And if these are patients who need to be tube fed or have tracheostomies or things like that. So you have to take all of that into consideration. As you heard earlier, the natural history may be unclear. And if it's unclear, it's really hard to define what are going to be your outcome measures, your endpoints, and how do you define an efficient clinical trial design? And I'm sure you've all heard that it wasn't the drug that failed, it was the design of the trial that failed. And that's really a letdown to the whole community. So part of our joint responsibility is to try to make sure that we're defining and developing efficient clinical trial design. And we're promoting inclusion as well, as you heard from others. So there's some families that have limited resources. They just cannot participate in the clinical trial even if they want. So how can we try to support them appropriately to get full engagement and access? And then if these are these ultra-rare diseases, there just may not simply be enough patients to get pharma interested. It may not be commercially viable to get an ROI. But how do you can get their interest, but sometimes the first phase 1 study or the proof of concept has to be done at an academic institution. It has to be done with philanthropic funds. And then at some point, maybe you can engage with pharma. And then as you heard earlier, ideally you want to have an internal control group. You want a placebo or a sham arm. But with an ultra-rare or rare disease, that's not always possible. Okay, there aren't enough patients to do a randomized full study. So can you use an external control group? And is it even ethical to have a control arm if you really have strong preclinical supportive data that the disease works. I mean, do you really want to disadvantage some of the patients that are randomized to the control arm? So these are all real-world issues that have come up. Okay, so in summary, you can provide the patient voice, you can help by advocating it, and you can support at these different levels. How much time do we got? We have to stop really soon, right? 9:45? A 15-minute break. Okay, I'll use a few minutes. Okay. Okay, so you want to make, you have to really try to understand your patient population. You need to work with the pharmaceutical company or whoever's designing the trial to try to identify a homogeneous group, because the smaller the population, the bigger the effect size you need to really convincingly say that the drug works. Okay? So there have been drugs that have been approved, I'll say for spinal muscular atrophy, a gene therapy approved basically on 12 patients. But the effect size was so fantastic that the grandmother could say, hey, that drug worked. You don't need to power this. You don't, okay, that drug works. And the FDA agreed, everyone agreed, that drug got approved. But that's not always the case. Now, in general, the FDA likes to have a confirmatory study. They want 2 well-conducted studies. What if you're using all the patients you have in the one study to do one well-conducted study? Where do you have the patients for the second? Well, they may not be there. So you have to work with the FDA, and they're very reasonable in my discussions with them. They understand the situation, but you need to go in early and have these discussions. You can't go in late to the FDA and say, well, here's our plan, because that's not the right way to get to success. So think about this, all of these topics in advance. Okay. Briefly, informed consent. So with what we're talking about here, the larger the magnitude of the disease, we're talking about generally catastrophic diseases with high unmet need. So in general, the patients and parents are willing to accept a higher risk than if it was a mild disease. Let's say you had eczema, which is, it's a bad disease, but it's not the same thing as having some of what you've heard about today. But the FDA doesn't always look at that. That you still have to show that there's a risk-benefit ratio, the equipoise. And so you need to make sure that you're identifying this, and that's where the focus groups come in and other aspects that you can help identify. I think it's also important that it's made clear that participation in a clinical trial is voluntary, that clinical trials are experiments. You're using an investigational product. It's not a treatment. But on the other hand, the parents are desperate. The patients are desperate. They see it as an access to treatment. So there's really a challenge here to say, yes, this is a potential treatment for your condition or your child's condition. But remember, this is an experiment. There's no promise of safety. There's no promise of benefit. And you may get randomized to a control arm. Quickly, accelerated approval. You've heard about this earlier. This is where a drug gets approved on the basis of a biomarker. You haven't really shown convincingly that the drug works. It doesn't improve things clinically. But the biomarker is felt to be so supportive of a clinical response. And I've put here the FDA language. It's reasonably likely to predict a clinically meaningful response, but there's a little bit of subjectivity there, how you interpret that. Okay, and pharma likes this because it's a quicker way to get to market, but it is a limited approval, and you're expected to, almost required, to do a confirmatory proper phase 3 study, but in a rare disease, again, you may not have enough patients, and it may take a while to do that. But for these rare diseases, it is a way to get to market earlier and help the broader population of patients suffering with that condition. Full approval is a higher bar. You need to have show clinical efficacy. Typically, you want to have 2 convincing studies, so maybe a small and then a big one, but you need 2 typically. And the use of control groups is something that's under discussion. I'm going to finish with single patient treatments. So there are evolving technologies such as these antisense oligonucleotides, ASOs, some cases small molecule drugs, and also gene therapy, all of which are being looked at to treat a single patient or a small number of patients with a particular genetic mutation. Okay. So single patient treatments have been around for a while by the FDA under the guise of compassionate use. And I'm not going to go through that here, other than to say there has been a pathway that's existed for a while. But it's now being kind of repurposed for these N of 1 personalized medicine programs. Okay, and I've just listed here a few of these that are currently up and running. NLORM, which is a philanthropic initiative, the N1 Collaborative, an academic initiative, 1M in Germany. So these are evolving. But the important thing is you may think there's only a single patient, but you have to think there's always a second patient out there with that same mutation. So what's N of 1 becomes N of 2, and then maybe N of 5, and at a certain point, the FDA is going to say, wait a minute, you need to do a clinical trial. And there's a gray zone here. So there aren't really guidances yet to fully help us figure out this pathway. And there are a lot of issues that I've listed here that I won't go into for the sake of time. There's some ethical issues. Who gets to be first? How do you select? So there's different aspects where, again, a patient advocacy group can help. Okay. So I'm going to stop there, and I'd be delighted to speak with you further at the coffee break if you have any other questions. But thank you so much for this opportunity and for your attention.
Thank you all for coming to the symposium, and thank you so much to our 4 speakers. This was a really wonderful session on research readiness for rare diseases. Thank you all.